Microbial Insights website within the next 2 weeks. A link will also be emailed to everyone who registered for the webinar. And at the end of the session today, just like all the webinars, we're going to have a short question and answer period. So feel free to go ahead and type your questions into the GoToWebinar software throughout the presentation. And then at the end, Frank will go through and answer as many of those as possible. So without any further ado, I'll go ahead and turn it over to you, Frank.
Dora, thank you very much. So hello everybody again. I have to apologize, my voice is not the best because I'm recovering from a cold, but I try to get through this as good as I can. So this is part 2 of the discussion of molecular biology for groundwater scientists, and I give a brief recap over the materials we discussed last week. So we talked about molecular biological tools. I introduced the concept and what those tools actually are. We talked about biomarkers, and we distinguished organism from process-specific biomarkers. We talked about DNA, the genes. We talked about RNA, the transcripts or the messenger RNA, mRNA, and the proteins. The proteins, typically the enzymes responsible for the reaction of interest, so the catalysts. We talked about quantitative polymerase chain reaction, qPCR, that can be used to quantify genes and also transcripts. We briefly discussed the relevance and importance for collecting and handling groundwater samples properly, because that's key in order to get access to the biomarkers. And then I mentioned that we currently apply an empirical approach to bioremediation. And the goal is to take advantage of the knowledge generated in academic institutions and advance the approach to do precision bioremediation. And I talk a little bit more about this today. So today we are going to talk about proteomic tools. So we look specifically at the enzymes that catalyze the reactions we are interested in. We talk about metagenomics and metatranscriptomics as approaches to gain understanding of the microbiology in an aquifer. And I will briefly discuss what these data actually mean and what do we really need to know to make meaningful decisions for remediation. And that then again comes back to my goal to introduce precision bioremediation. So we talked about the central dogma in biology. You have DNA, and then the flow of information goes to the RNA and to the protein. And so the DNA can tell us who is there and informs us about potential activity. The transcripts, the mRNA, tells us who is active, what genes are transcribed, and gives us information about the actual activity. And then the protein tell us what functions are actually expressed by these microbes. It informs us about actual activity. So this is certainly a very good biomarker because it's actual activity and not just potential activity, what we can gain from DNA analysis. Now the tools to get information about these different types of biomarkers are listed on the right-hand side. We talked about qPCR, and today I'm going to talk a little bit about metagenomics and amplicon sequencing as tools to gain information about the DNA. The tool to get quantitative information about the RNA is in principle qPCR, but you have to have an additional step that's called reverse transcription. You use a specific approach to transcribe the RNA into DNA, and then you quantify the DNA. This is detail. Just realize that we can quantify RNA. And now to proteomics. This is the first topic I want to discuss. How can we measure proteins of interest in environmental samples? So before I start doing this, I just want to make something clear to everybody. When we look at nucleic acids like DNA and RNA, we extract these macromolecules from an environmental sample, we have very, very little. So there's very little DNA and very little RNA in the sample. So the example here I have, we have two target molecules of a gene that we are interested in, and there's very little in the sample. So we apply PCR. And in the first round of PCR, these two molecules are duplicated. You get 4 copies. In the next round of PCR, you get 8 copies. And after 30 rounds of PCR, you have more than 1 billion copies. So out of 2 copies of a target gene, you made 1 billion. What that means, now we have enough material to analyze it. Okay? So this chain reaction generates copies from copies, and we get enough material that we can analyze. And this can be done in a quantitative fashion. Now for proteins, this is fundamentally different because we cannot amplify protein. So whatever the amount of protein is in your sample, that's all you have. Okay? There's no way to amplify protein. So that's a fundamental difference. So now you may wonder, can we really quantify or detect protein in environmental samples? And the answer is yes, we can. Because the technology to detect proteins and peptides has advanced so much that the sensitivity of these high-end instruments is so good that we can get away without amplification of protein. We can still detect it. Walk you through the process real quickly. So we collect the sample, in our case a groundwater sample. Then we collect the proteins. There are standard procedures out there, just like DNA extraction, we extract protein. The proteins are lysed, the cells are lysed and the proteins are digested with trypsin. So we get peptides. Each protein is cleaved into smaller pieces, so-called peptides. Then we use a liquid chromatography system to separate groups of peptides from each other. But all of the peptides go through this process. And as they come out of the column of this liquid chromatography system, they are going into a high-end mass spectrometer. And this mass spectrometer sees every peptide that's going in it. So all the peptides from all the proteins in your sample are being analyzed. And you get a large amount of raw mass spec data. And then there's a computational pipeline that helps us identify and assign these different peptides to specific genes and proteins that can then be recognized. So for example, we would see all of the D-glucocoides proteins that were present in the sample that was collected from a groundwater. Okay, so this is in principle the process. What I just described is called global proteomics because you look at all the proteins in a sample. What is critical for this approach is a good database because you can only identify the proteins if you have the gene sequences available to match the sequences. Otherwise, you cannot identify or assign the peptides to a certain protein. Now you can imagine this is a complicated approach and you get a lot of information, much more than you actually need. And it takes a long time. Each run takes about 24 hours. So it's not a high throughput technique. But we can make this simpler. And this is shown on this slide. So we don't look for all proteins in a sample. We just look for those that we are interested in. Let's say reductive dehalogenases that are being made by the microbes in the groundwater. So then we apply a different approach, and all the proteins that go into this mass spectrometer, we don't look at all of them. We have a gate and block all the ones we are not interested in. And we only look at the reductive dehalogenase proteins, the green ones, and get mass spectral information from those proteins. So we ignore most of the proteins. We only look for the ones that we are interested in. And then the mass spectrometer gives us a signal for the proteins that we select for what we want to see, only reductive dehalogenases. And we can include in this approach a known amount of this particular protein of interest. What that means, we can get quantitative results. We can quantify how much of our target protein is in the original sample. This approach is simpler. This mass spectrometer is a simpler instrument. It's already available in many analytical laboratories and can be used for this approach. So the targeted approach, this is how this is called, targeted proteomics approach, that's probably the approach that's of greatest interest for monitoring proteins of interest in environmental samples. So I just here compare real quick these two approaches. With global proteomics, we look at all proteins. With targeted proteomics, we only look at specific proteins of interest. Here we need high-end instrumentation, more expensive, more complicated. Here we can use simple mass spectrometers. Here this is more computationally intensive. Here we can say this would be a routine analysis. In global proteomics, we generally only get relative quantification. We can say protein A is more abundant than protein B. With targeted proteomics, we can achieve absolute quantification. I mentioned this last time, the difference between an open approach, so we look at everything that's there. In a closed approach, we only look for the proteins that we are interested in. If there are other proteins that could be of interest, we will not see them unless we include them in the analysis from the beginning. I talked a little bit about the database requirement, and this is important. And for the targeted proteomics approach, there's also a little issue with allelic variation, and I explain this real quick. The targeted proteomics approach, for example, you target VCRA. That's an enzyme that converts vinyl chloride to ethene. This enzyme has 1,500. The gene has 1,560 base pairs and encodes 519 amino acid long protein. Now, not all of these VCRA sequences are identical, but they have the same function. Okay? So now this is just an example from a blast analysis to a publicly available database. Looks complicated, but basically what this database lists are all the VCRA reductive dehalogenase genes, and you see here the percent identity of these sequences. The point is they are not all 100% identical. So some are only 97% identical. If you look at what this means, it means that up to 16 amino acids are different. But the protein is still VCRA and has the same function. But now if a single amino acid is different in one of the peptides that we analyze in proteomics, you will not see it because the database doesn't show this variation. So you don't see the peptide, you would conclude the protein is not present. You get a false negative result. This is a little complicated, but it basically means we have to know the entire sequence space of a particular gene in order to make sure that we would capture all the proteins encoded by this gene in an environmental sample. For the global proteomics approach, in order to identify the proteins, all the peptide sequences must be matched to the gene sequences. So how do you get all the gene sequences in your sample? There are two options. So people try to assemble a generic database. They just make assumptions what types of genes are in your matrix, in your aquifer, and use this database to match the peptide sequences that can work, but it's not ideal. A better approach would be that you actually determine all of the sequences that are in your aquifer. Okay, you determine all of the genes that are present, and then it's very easy to match all the peptides. This can obviously also be applied to the targeted proteomics approach. So this all was a little complicated. The bottom line is proteomics only works well if you have a good understanding about the gene sequences that encode for your proteins of interest. And in order to get this information, we have a very good approach called metagenomics. So metagenomics can make the proteomics approach much more meaningful. And now I'm going to talk briefly about metagenomics. In metagenomics, you analyze the genomic DNA of every member of the community. Okay? This term was first introduced by Joe Handelsman in 1998. So you take an environmental sample, you extract the DNA, and you analyze all the genes representing every member of this community. So you take the DNA from the community, basically your groundwater sample, you collect the biomass, extract all DNA, and you sequence all of this DNA, and then you get many sequence reads. And then you do a computational analysis, bioinformatics. And when you do this right, what you get out of this, you get an information about the entire genetic potential of the community, all the genes that are there. You can do the same thing for the RNA. So you get the mRNA from the entire community. The only difference is you don't extract DNA, you extract RNA, you sequence everything, and the result tells you all the active genes of the community. This can be done. Now, I give you a variation that you probably makes it easier for you to recognize what the power of this approach is. You take the community, you extract all DNA, you sequence all of this DNA, you do your computational analysis, but now instead of looking at everything, all the sequences, all the genes in this metagenome, you specifically look for a subset of genes of interest. So for example, you can identify all of the 16S rRNA gene sequences. You can assemble these genes, and then you can perform a phylogenetic analysis. And that tells you who is there. You can now tell what types of organisms are present in my sample. And then obviously you would see in an aquifer that has Dihalococoides, you would determine that Dihalococoides 16S rRNA genes are present. You can also use the same rationale, but you ask the question, what reductive dehalogenase genes are in my sample? And you can extract all of the reductive dehalogenase genes in your sample. Okay, so then you have some information about the potential to do reductive dechlorination that is present in your sample. So the key point is you don't have to look at all the information. You can down select and only get information that is relevant for the purpose of your remedy. So another thing I would like you to understand is the difference between shotgun metagenomics. This is what I just discussed. You sequence everything, a shotgun approach, compared to amplicon sequencing, which is a more targeted metagenomic approach. I briefly explained this. So here's your sample, the biomass you extract from your groundwater. There are many different organisms in there, including our warrior right organism of interest. Example, Dihalococoides to do reductive dechlorination. You extract the DNA from all of these, from the entire biomass. And then you get the genomes of all of these different organs, representing all of these different members of the community. Then you sequence everything, high throughput sequencing. This is a rather complicated technique. You don't need to know the details. All you need to know is that there are many places where you can get sequencing done. So I know Microbial Insights, for example, now has this capability, and you can ask them to sequence all the DNA in a given sample. So this is relatively affordable. The prices have come down significantly, and it's a routine procedure. What you get are many sequence reads, and the challenge then is to put them all together. This is the bioinformatics aspect of high throughput sequencing. And when this is done properly, you get the entire genetic complement of all community members. Basically the genetic potential, what I mentioned before. Then you can extract sequences representing your target gene. In the case of the 16S rRNA gene, you get this information, who is there. Again, that's the shotgun metagenomics approach. A variation of this approach is you don't sequence everything. You do an amplification with this community DNA using primers targeting a specific gene of interest. Let's assume it's the 16S rRNA gene. Then you generate amplicons of this particular gene, so PCR products of amplified 16S rRNA genes of every member in this community. And then you apply the high throughput sequencing approach. So what you get sequences of these amplicons that were amplified in the specific PCR reaction. That gives you the same information, or should give you the same information. Who is there? So this approach is called amplicon sequencing approach. So it's also a type of metagenomic approach because you look at the entire community, but then you down select and only look for a specific gene of interest. So be aware there are shotgun metagenomic approaches and amplicon sequencing approaches that can be applied. Here you get a lot more information, but then you can down select using bioinformatics. Here you apply a more targeted approach and you get only information about a particular gene of interest from the entire genetic material that's present. But in the end, you may get similar information that you would like to have or need in order to make decisions. Both approaches are doable. I give you here an example of an 16S rRNA gene amplicon sequencing approach. But if we would have done a shotgun metagenomics approach, we would have gotten the same type of information. I just want to point out what type of information are we actually getting. So this is an enrichment culture. It's not groundwater. So the diversity of different types of organisms is a little bit lower here than what you would expect to get from a groundwater. But the principles I explain now are the same. Here's the inoculum. And this is a culture that degrades dichloromethane. So this is the distribution of different types of organisms in the inoculum. When we grow this culture with dichloromethane, you see that the red bar here increases significantly. This is the DCM degrading population. So this is responding to the presence of DCM. The organism takes advantage of DCM as a growth supporting substrate, and the DCM degrader is growing. The same culture that doesn't receive DCM, here no DCM, you see that the red, the DCM degrader, cannot grow. In fact, if you compare the size of these bars, it's declining. If we feed this culture hydrogen, you also don't see the DCM degrader increasing in abundance. So this is the type of information we can get from amplicon sequencing and looking at the abundance, the relative abundance of 16S rRNA gene sequences. And now this is what I would like to point out, very important. We get relative abundance information. So we can, for example, say, in this example here, the DCM degrader is the most abundant population and makes up probably 70% of the entire community. And this orange population has a relatively lower abundance. We can also compare to the previous the inoculum, and we can say the relative abundance of the orange population is decreasing. We can say the relative abundance of the red population is increasing. What we don't get is absolute quantitative information. I cannot tell you from this data set how many of these DCM degraders are present. I can only tell you it's more than 70% of the entire community, but I don't have an absolute number. So this is one of the shortcomings of these approaches, metagenomics and amplicon sequencing. We don't get absolute abundance information. Theoretically, these approaches could provide absolute abundance information, but not at the current time. So theoretically they could, but that has not been worked out in detail. So this is a big difference between quantitative PCR and amplicon or shotgun metagenomics. You have to understand that relative abundance information has value, but it cannot replace quantitative, absolute quantitative information. Now I'm going to give you a brief outlook what can be done at the moment, that can be done now. And I call this site-tailored application. So you have your site, you collect the groundwater sample, and you do metagenomics. Then you know all of the genes that are present in your microbial community, present in your groundwater of interest. Then you can, for example, ask the question, what reductive dehalogenase genes are present at your site? So you don't just ask, is VCRA present? You ask very specifically, what type of VCRA and what other reductive dehalogenase genes are present at my site? You can also, since you have everything in the metagenome, you can also ask other questions. For example, what genes implicated in petroleum hydrocarbon degradation are present at my site? So you can ask very specific questions. And now, with this information, you can design qPCR approaches that specifically target those genes of interest that are actually present at your site. You can obviously then also target the transcripts of these genes. And now, since you know what genes are present, you can apply a targeted proteomics approach that is not biased by allelic variations, because you know exactly what type of VCRA or any other reductive dehalogenase protein is potentially expressed or present at your site. So then there's no bias. And now you can take this information, do an integrated analysis, ideally together with geochemical, hydrological information. And I argue that if this is done properly, you can generate predictive understanding and implement more efficient treatment at your site. So this is just a conceptual figure that you've probably seen before about plume treatment. What we do when we do in situ bioremediation, we often install bioreactive barriers. So we have a source zone or a plume or a source zone, and that generates a plume. And the plume intersects a bioreactive barrier where we can do biostimulation or, if necessary, bioaugmentation. And ideally, what gets out of this reactive barrier is clean groundwater. So now, what has been happening a lot is biostimulation with electron donor is often key to stimulate the Helicoccoides, Halogenemonas, and other organisms involved in reductive dechlorination. What we often forget is if we biostimulate too much, there are negative consequences, secondary impacts of biostimulation on aquifer groundwater quality. For example, the pH can go down, which has negative impacts also on reductive dechlorination activity, because these Helicoccoides, the Halogenemonas organisms, they don't like pH below 6 or 5. They stop doing anything at pH 5.5. The pH reduction can also result in the dissolution of toxic metals. That would impact groundwater quality. You get excessive methane formation. You have high DOC, organic acids, and you may generate solids that actually then influence aquifer permeability. So what I'm trying to say is too much biostimulation can have negative effects. The point being, if you have a better understanding about the needs and requirements of the microbiology that catalyzes the key steps in detoxification, you may be able to biostimulate less and get, in the end, a better result. So this is one example what I mean with precision bioremediation. So often I'm asked, Frank, can molecular biological tools give us rate information, in situ rate information? It's kind of the holy grail of engineering a contaminated subsurface environment. So here are our little warriors, these reductively dechlorinating populations. And now I hope it's clear now that we can quantify DNA, RNA, and protein biomarkers. And in theory, right, those biomarkers should correlate with the activity of a particular organism. The question then becomes, can we generate robust rate estimates based on quantitative biomarker analysis? Right, so this is a difficult task to determine in situ dechlorination rates. And so my point I want to make is, I believe there's an opportunity to use these quantitative measurements and extract rate estimates that you can expect to see in your aquifer. If this is then integrated with geochemical, hydrological information, could include co-contaminants and other things one could measure, and you take this quantitative biomarker analysis, I believe there's an opportunity to get reasonable in situ rate estimates. We are not quite there yet, but I believe in the near future, there will be work being done that explores and possibly validates this type of approach. And then obviously, if you have reasonable rate estimates, you can project the longevity of your plume. And this will also help people to argue for site closure, because you have a justifiable argument that the rates that are expected will take care of the contaminants in a certain period of time. Okay, so the key point, the analysis that includes metagenomics, metatranscriptomics, and proteomics can eventually lead to reasonable in situ rate estimates with obvious importance for remediation and site closure. Okay, so I argued that at the moment we do empirical bioremediation, but with all the knowledge that has been generated or academic institutions are currently generating, we can move this empirical approach to a scientific approach with predictable outcomes, what I call precision bioremediation. And this will ultimately lead to a greener technology to apply biostimulation, bioaugmentation, and probably also reduce costs and have more desirable outcomes, not just for destruction of the key contaminants, but also reducing secondary impacts on groundwater quality. So this is, I don't talk as much today than I did last time, but I want to point out before I come to a conclusion that there's a meeting coming up, the Battelle meeting in Miami, Florida in May, and Carmen Lebron and myself, we are chairing a session called Next Generation MBTs, a pathway to precision bioremediation. And there we invited some key people in the areas of metagenomics, metatranscriptomics, and also in the area of metabolomics, who can probably much better than I did today explain to you how these technologies can lead to a better understanding of the microbiology and ultimately improve bioremediation approaches. So metabolomics is an approach where we look at all of the metabolites in a given sample. And you can imagine there are lots of different compounds as metabolites in these different cells and in the aquifer matrix itself. And analyzing all of those can give us information about the physiological status of the microbiology. And this can also be a key component in this integrated analysis. So I hope I could give you some idea where molecular biological tools are moving, and we haven't reached the end point yet. There's still a lot to come that can ultimately make aquifer remediation a more predictable business. So again, I have to thank the key funding agencies, SERDAP and ESTCP, Microbial Insights, for putting this webinar series together, and then many of my students, colleagues, and collaborators. And then I also have a greeting to you from the key microbiology. They all say Happy Valentine's Day. And with that, I'm going to end. And if there are questions, I'll be happy to answer them.
All right. Thank you very much, Frank. That was a very informative webinar, as always. And we do have several questions that are started. Again, if you have questions, please go ahead and type them into the GoToWebinar software, and we'll answer them, as many of them as possible. Okay, the first question that we have, Frank, is about catalytic proteins that can be manufactured by bacteria extracellularly. Do you worry about seeing those as free molecules in the groundwater?
I'm not sure why I should worry about this. So for reductive dechlorination, those proteins are not extracellular. We know exactly where they are located. We know a lot about the biochemistry, and they are associated with the cells. Now there are other processes that can be mediated or catalyzed by extracellular proteins. So this is an interesting question, because if you collect the biomass and then extract the proteins from the biomass and do proteomics, you will obviously not see the extracellular proteins that are not collected together with the biomass. So then you have to if you know that your protein of interest is an extracellular protein, this will be on the outside of the cells. So then collecting the cells, you will obviously not collect the protein. Then you have to apply a different approach. And that's probably something that can be done, but I have not encountered that yet. But I think it's certainly doable. Did that answer the question?
Yes, I think so. Okay, the next question, and maybe we want to broaden this a little bit, but he's asking how much sample is required from a well or geoprobe location to conduct a bench scale study. Maybe we can also talk about how much from a monitor, or I guess for a qPCR study as well, since some people may not be familiar with that, or the proteomics approach. And then I have a second part to that question after you answer that.
Okay, so how much groundwater do you have to collect in order to do these different types of analysis? So I pointed that out in my talk today, that for a qPCR-based analysis, you need very little DNA because you have this big amplification step. For metagenomics, what you would like to have is enough DNA that is representative of the entire microbial community in your sample or in your environment of interest. In general, a liter of groundwater would give you, in most cases, enough DNA to do these types of analysis. The same can be said for the RNA. But RNA, I mentioned this last time, is more complicated because it's always prone to degradation. So you have to take precautions there in order to get a representative picture of the mRNA. Now, for the protein, it can be a little bit more difficult. I mentioned that you cannot amplify protein, but the instrumentation is very sensitive. So we did several studies with environmental samples, and what we observed is that you do need more groundwater for proteomics analysis if the populations that harbor the protein of interest are present in low abundance. So for example, if you're looking for a reductive dehalogenase protein of D. leukocoides and you only have 10 to the 3 cells per liter, a liter of groundwater is not sufficient to do proteomics. Then you would need a larger volume. If you have 10 to the 6 cells of D. leukocoides per liter, a liter of groundwater is plenty in order to detect reductive dehalogenases of D. leukocoides. So we are on the, or not we, I should say, researchers on the process developing exact guidelines of how much sample do you need to collect in order to perform a meaningful proteomics analysis. And this can be based on initial qPCR results, where you quantify how many of your organisms of interest are present. And then you can judge, based on that result, how much groundwater should be collected so that I have enough protein to do a meaningful proteomics analysis.
So kind of doing a tiered approach in the analysis, starting with the qPCR, then moving to the proteomics?
Right. So for many sites, the initial qPCR may not be needed because you already have this information, most likely. But if you don't have it, I would recommend first doing a qPCR analysis that you get an understanding how many of your target organisms are present. And then you can gauge how much groundwater do I need to collect to get enough biomass to do a meaningful proteomics analysis. You don't want to spend money on proteomics analysis if you don't have enough of your protein present, right? So it's kind of a safety. You can check and make sure that the money is meaningfully invested and you get positive results back. You don't want to do a proteomics analysis and then people tell you we couldn't detect anything. That's why I'm saying it's important to make sure you have enough material present in your groundwater. biomass, I mean.
Okay. The second part of that question is, how do you feel about a grab sample? Is that more or less representative than a sample from a developed or purged monitoring well?
So a grab sample, that means you look at solids, right?
Mm-hmm.
So most of our efforts have focused on groundwater because we recognize that getting solid samples at many sites is too expensive, difficult, or impossible. But groundwater samples can typically be collected because you always have monitoring wells to extract groundwater. So that's why everything or most things we do are focused on groundwater. Now, of course, it's not a bad thing to get solid samples and do analysis with solid samples. That can be done. So you can extract DNA, RNA, and proteins from solid samples fine and do this type of analysis. Now, there's always this question then, how many of the organisms or the biomarkers of interest are associated with the solids, and how many are associated with the groundwater? And this is always can be a challenging question. I gave you a literature citation last time. Natalie Capiro and did a nice study where she analyzed how many D-leukocoides cells are in the groundwater, how many are attached under different conditions, so different soils, different properties, different conditions for the D-leukocoides cells. And there can be a change in distribution, and that obviously is important for judging the total amount of D-leukocoides in the aquifer, right? So we have a pretty good understanding how this works for groundwater. I'm a little reluctant to say I have a good understanding. If you take a grab sample and just analyze the solids, I would have a hard time figuring out how many D-leukocoides cells are really in the aquifer formation. So again, it can be done, ideally then together with groundwater analysis. But I think for the majority of sites, I think it's justified to focus on groundwater only.
Okay. One of the concerns with the new analysis like proteomics is, how do we report unknowns? With DNA and qPCR, we have to know the gene sequences for identification and quantification. So how are we handling unknown proteins? How are these going to be reported? And how are you going to get a sense for the amount of unknowns that are present in the sample?
So this is a major challenge. So if we do metagenomics, or make it even simpler, you have a D-leukocoides genome. About 30 to 35% of all the genes on that genome, we have no idea what these genes are responsible for. We don't know what they're doing. We don't know what the proteins that these genes encode, what their function is. When we do a metagenome, right, the numbers don't change. 30% of all the genes that we identify, we have no idea what they are doing. So if we detect the protein, we know that this protein does something because it's there, but we don't know what it does. So, I mean, I don't want to be cynical, but just ignore it, because if we don't know a function, you cannot make any conclusion. This is a major issue and has to be addressed from a scientific point of view. People have to spend more time doing biochemistry rather than just identifying genes and never spend any time figuring out what their function is. So this is an issue, but the glass is half full in my opinion. When I say 30% have unknown function, that also means that 70% have known functions. And it's certainly a good approach to focus on those that have known functions and make meaningful conclusions based on what we know, and don't torpedo this approach by emphasizing that 30% are unknown. So we know a lot, but we don't know everything.
Okay. I'm not sure that I completely understand this question, so if the person that asked this, if this isn't clear, you may have to clarify what your intention is for this. But the question reads, Frank, is the biomass in the aquifer 100% bacteria? I'm not sure if they're asking if there's like archaeal and fungal and other organisms that you would expect to be present.
Oh, no, obviously not. So there are other microorganisms in the aquifer, right? There are fungi, there are protists, there are archaea, there are bacteria, right? So there is other DNA in the aquifer. So when we do a metagenomic analysis, we would also get sequences from other organisms, including viruses, right, that are not just bacterial DNA. Then it depends what we do with the analysis. We can identify these gene sequences that belong to an amoeba or some other organism. We know that these organisms are not directly involved in the processes that we are interested in. So then we can ignore them by doing the bioinformatics analysis. But theoretically, we could also look at those organisms and their genes and their proteins. But this would only make sense if there's evidence that these types of organisms play a role in the process of interest. So metagenomics, we also get information about other organisms that are in the sample, not just bacteria.
Okay. The next question is, do you have SOPs that are available for groundwater collection?
So I mentioned that last time. The key thing for groundwater collection is when you start using a particular procedure, don't change it over time, so that all your samples you collect from the same well can be directly compared. In general, we had better experience with low flow sampling techniques. And I think I made 2 publications available last time that describe an approach that we developed together with geosyntech researchers, where we tested different types of groundwater collection methodologies and then did biomarker analysis and then just asked the question, which one is most reproducible? And that was definitely a low flow sampling technique, and the details, the SOPs, how this should be done, is written in these papers. And I thought, Dora, I sent those to Microbial Insight.
Yes, those are available if anybody needs those. I think Anita sent the links out for those in the last webinar document.
So there may
the other thing.
I'm not saying you have to use those. There are probably other opinions out there. And so I'm not saying that this is what you have to use in order to make this analysis meaningful. This is something that we recommend. But there are others out there. The key thing is don't switch between different methodologies. Stick with the one you're using so that the data you generate over time are comparable.
We also have some SOPs that are available for sample collection on the resource section of the website, and there's videos too that can show you how to use the filters and how to do groundwater collection, what we would recommend for bottles as well, so you can clearly see how this would work in the field. Okay, the next question, Frank, is how expensive are these tools to apply? Is it practicable or feasible to do these at larger sites, like if you had a half-acre or acre site?
So the size of what the size doesn't matter. What matters is how many samples do you collect from that site. So, I can't give you exact prices. So, for example, the proteomics analysis, to my knowledge, is not yet commercially available. What is commercially available is metagenomics analysis and also metatranscriptomic analysis and obviously qPCR analysis. For the metagenomic analysis, the prices have come down. I mean, it always gets cheaper because the sequencing technology comes down in price significantly. You get more and more information at lower price. I don't want to give you a number, but I give you some idea. If you submit one sample, it's probably more expensive per sample than if you submit 10 samples. What else can I say about this? There are commercial places that do this type of sequencing. And you can just do a Google search, sequence analysis and commercial. You will find places. They give you dollar amounts for a certain type of service. And you will notice that the numbers between different providers don't match up at all. And I think they do this on purpose. So the entire analysis requires different steps. And they often tell you this step is this much, and the other provider tells you this and this and this step is this much. So then the numbers don't match. But the total number that you will be charged is probably approximately the same. Okay? So this is a little bit tricky business. I would estimate. So let me do one more thing here. So I have a sequencing machine in my laboratory. So we can, in order to sequence, we have to buy certain chemicals, kit from the company. That kit costs $1,200. Now with this kit, I can sample 10 samples or 20 samples. But now if somebody only wants one sample analyzed, I still have to buy this kit, right? And I would have to charge the entire amount for one sample. If I had 10 or 20 samples that I could run with one kit, obviously the price per sample would come down. So therefore, it's just a little tricky to answer the question about cost. I think once more people ask for this type of service and the analytical laboratory gets enough business, the prices will come down further because you can use one kit for multiple samples. The bottom line to me is that this type of analysis can provide a lot of information and the cost is not prohibitive. So I think there are lots of opportunities to take advantage of this technology and make remediation, advanced remediation. I think don't be afraid of the cost.
That's what I was going to say is I think that it's important for people to remember qPCR is routinely used and has been now in site assessment even on very large projects for more than a decade. So I can speak from Microbial Insights perspective. You're talking around $275 for a target, and then if you do additional analysis of organisms or functional genes, then it gets cheaper because we have that DNA extracted on the sample. And we're happy to provide any of that information that someone would like. For next generation sequencing, most people are doing the amplicon sequencing right now commercially is what's primarily available. And the cost on that between companies typically ranges from what I've seen from $600 up to $1,000 for samples, and that's if they can batch it with other samples. So your turnaround time may be a little slower because, like Frank was talking about, you got to get a group of samples to run together to make the cost not prohibitive in those situations. So if anyone has questions about that, you can contact either me or Frank, and we can provide you some additional information on that. I hope that's okay, Frank, that I helped. from our
That's
end.
fine. That's fine. Okay. Yeah.
Okay, so the next question, with qPCR, are you concerned about introducing errors or mutations because of the sensitivity and doing so much amplification?
Can you repeat that question? I didn't.
It says, with qPCR, are you concerned about having errors or mutations that are introduced because of the sensitivity of the assays and we're doing 30x or more amplification?
So, I mean, there's a lot of literature about PCR bias and what can happen and what does not happen and how this would influence the results. With everything I know, this is not a concern. I also have to say that 30 cycles in my example, that's probably on the high end. So most qPCR reactions would not require that many cycles. So the chance of, errors accumulating and giving you funny results is very unlikely. So I don't think that this is from a practical point of view, a concern that we should worry about. There's enough literature and, experiments have been done to confirm that this is not something we need to worry about when we do qPCR analysis. You have to recognize when we do TACMA and qPCR, we only amplify a very short piece of the gene of interest. And if there are errors that would affect that region, the probe wouldn't bind, you wouldn't get a signal. So this is very unlikely that this would affect the outcome of the analysis.
Okay. Do you see these techniques and treatments being applied to waste before they enter the environment? And if that's being done, do you think it can be done effectively?
Okay, that I didn't understand. Say that again.
Sure. It says, are these techniques and treatments being applied to wastes before they enter the environment? And if so, can they be done effectively?
To wastes before they enter the environment. So I'm not entirely sure. So waste. What type of waste? So we're not talking about chlorinated solvents then. We talk about other waste streams.
Yeah, it doesn't specify.
I'm not entirely sure how to respond to this. It depends on what waste streams you're talking about. So obviously, if these chlorinated solvents were released in most places in the United States many decades ago, right? So you can't go back and look at the waste stream. Yeah, I'm not sure what to say about, but if the person can specify the meaning of waste stream a little bit more and email me directly, I will try to answer if I can.
Okay. The next question is, have you implemented any of these analytical processes to wastewater for targeting alternative contaminant removal?
Well, so we certainly attempt, whenever we work with a contaminant, the question always comes up, can we generate biomarkers for monitoring a population or a specific process biomarker so that we gain more understanding. So we're trying to do this. But the focus has clearly been on chlorinated solvents and reductive dechlorination. Obviously, everything I talked about today and last week, the focus has been on groundwater remediation, but these tools clearly can be applied to any type of environment. So you can take samples from a wastewater treatment plant. You can take samples from contaminated soil. You can take samples from contaminated sediment. You name it. The same types of approaches can be applied to different environmental matrices, not just to groundwater. But then obviously you need to know a process. What are you looking for? what type of information would you like to get? Maybe something in general I would like to add, I should have said earlier. You know, metagenomics is sexy. Every student coming to my lab or to other professors' laboratories, they all want to do metagenomics, metatranscriptomics, because it's cool. But if you just do it, you get a bunch of sequences and a lot of information, and then you don't know what to do with it. So you do have to have a specific idea, goal in mind. This is what I want to know. And then ask the question, can metagenomics help me to get this information? And if you decide, or other people can help you making this decision, if metagenomics is an approach that can help you to get this type of information, then do it. But if you just believe, oh, let's do metagenomics because it's cool and everybody does it without having a specific question, then it's not necessarily meaningful. It can be still, but maybe not. So first, ask the question: what do I want? What do I want to get out of this analysis? And then decide on the appropriate tool.
I definitely agree. That's really important. You want to do that with any kind of experiment, right? Is figure out what question you're trying to answer before you move into it.
Right, right.
All right. Well, thank you, everyone, and thank you, Frank. I know we ran a little bit over time. I appreciate it. I know you're not feeling well, so I appreciate you extending your voice that extra few minutes to help us answer all the questions for everyone. And again, we have another series next week. I believe Dr. John Wilson will be presenting on that, and that Frank will be available as well for any questions that you have. But we hope you can join us again. And again, thank you so much, Frank, for this informative webinar series. I know it's been very beneficial, and we've had great feedback so far on it, so we appreciate your time to do this.
My pleasure. Thank you.
All right. Have a great day. Happy Valentine's Day, everyone. Thank you.
Bye-bye.
Bye-bye.